Imagine yourself lying in a calm, dimly lit space, where the world outside your door has softened into something distant and gentle. The noises of the day have faded to a low hum, like a tide retreating from the shore. Your breathing is slow, steady, and unhurried, and with each breath you take, you can almost sense the quiet rhythm of your body at work. This rhythm is not loud or demanding. It is patient, constant, and endlessly reliable, the music of life that has been playing within you since before you could remember. Beneath your skin, a vast community of organs, tissues, and cells is working together in quiet coordination. Each has a purpose, each knows its role. The heart keeps time with its steady beat, moving blood through countless vessels. The lungs expand and release, bringing in fresh oxygen and letting go of the carbon dioxide your cells no longer need. The digestive system moves in its own slow cycles, transforming food into energy. Muscles tense and relax in response to even the smallest adjustment in posture. The body is not a loose collection of separate machines. It is a network of living relationships. One system answers to the needs of another. A change in one place is felt elsewhere. A message sent through the nerves may ask the muscles to act, or it may call for the glands to release a hormone. The circulatory system delivers not only nutrients but also these chemical messengers, allowing one part of the body to speak with another. And within this harmonious arrangement is the reproductive system, a set of organs and processes unlike any other. It is different because it does not exist for your immediate survival. You could live your entire life without it ever producing new life. And yet, from a biological perspective, its work is deeply significant. It is the system that allows life to continue beyond your own years. It links you to the people who came before you and to the generations yet to be born. The human reproductive system is actually two systems, complementary in design, the male and the female. They are built differently, and they serve different roles, but their purposes are aligned. Each one is specialized to produce gametes, the cells that carry half of the genetic information needed to create a new human being. In the male system, these cells are sperm. In the female system, they are oocytes. The design of each reproductive system reflects its role. The male system focuses on producing large numbers of gametes and delivering them efficiently. The female system produces fewer gametes, but creates a supportive environment where fertilization can occur and, if it does, where a developing life can be safely nurtured. Even so, neither system works in isolation. Their functions depend on the cooperation of other organ systems. The endocrine system provides hormonal signals that control reproductive cycles and coordinate the timing of events. The nervous system relays information, triggering responses in muscles and glands. The circulatory system delivers oxygen and nutrients to reproductive tissues, ensuring they remain healthy and able to function. This interdependence is a reminder of the body's unity, that no process is entirely separate from the whole. You might imagine your own body as a living garden. In this garden, the reproductive system is a seasonal plant. It has times of growth, times of rest, and moments when it blooms. Its cycles are not constant, but they are reliable. In youth, it begins with a quiet preparation. As maturity arrives, the system enters its most active phase, producing gametes and responding to the rhythmic patterns of hormonal changes. Later, as age advances, the system slows, and eventually it rests again. This garden imagery helps us see that the reproductive system is not simply a set of organs with fixed roles, but a dynamic process. Adapting to the different stages of life, it responds to signals from within and from the environment around you. Stress, nutrition, temperature, and overall health can all influence its function. Yet even in stillness, when it is not actively producing gametes, the system remains a living part of you, connected to your body's overall balance. The beauty of this system lies not only in its ability to create new life, but also in the intricacy of its design, from the level of a single cell to the arrangement of whole organs. Every detail serves a purpose. The male reproductive system contains structures that regulate temperature to keep sperm healthy. The female system includes layers of tissue in the uterus that can transform themselves to welcome and support a developing embryo. These details are the result of countless generations of refinement through evolution, shaped to fulfill a shared purpose. As we begin this session together, we will move slowly through the reproductive systems, not rushing, not straining to remember every detail, but simply listening to their story. We will look closely at each structure and consider its role. We will notice how one part supports another, and how the whole system connects to the rest of the body. You may find that some of what you hear feels familiar, perhaps from school lessons or earlier reading. But here, the pace is slower. The explanations are softer, less like a test and more like a walk through a quiet museum. We will linger on the details that help you picture the system clearly in your mind. We will allow time for each image to settle before moving to the next. For now, there is nothing to remember or recite. You are simply observing, in the same way you might watch clouds drift across the sky or waves move along the shore. The human body is vast in its complexity, and the reproductive systems are just one part of that great landscape. But even this one part holds stories of origins, of cycles, of connections to the past and to the future. In the parts to come, we will trace the origins of the reproductive systems, beginning with the earliest stages of life, when male and female structures are not yet distinct. We will see how they develop, how they take on their specialized forms, and how they are guided by the chemistry of the endocrine system. We will visit each organ in turn, moving gently from one to the next, until the whole picture is clear. As you rest now, know that the same quiet rhythms we speak of are alive within you. The heart continues its steady beat, the lungs rise and fall, the cells carry out their patient tasks, and somewhere deep within, the reproductive system exists in its current season, perhaps in activity, perhaps at rest, but always connected to the greater harmony of your body. It is within that harmony that the story of reproduction begins, not as something apart, but as one chapter in the continuous narrative of life. And as we turn each page together, we will keep that thought close, that this system, with all its complexity and purpose, is part of you and part of the long and quiet continuity of human life. 2. Why reproduction exists in living things. Reproduction is one of the most fundamental characteristics of life. It is the quiet bridge between one generation and the next, the biological thread that connects you to every human who has lived before and every human who will live after. Without it, even the most vibrant and successful species would fade away in time. The individuals might live out their years, but the story would end with them. From the smallest single-celled organism drifting in a drop of water to the most complex mammals on land, reproduction ensures continuity. In its essence, it is the process by which living things create new individuals of their kind. This new life carries forward the genetic instructions that shape not only the body's structure and function, but also its potential to adapt, survive, and thrive. In humans, reproduction is sexual, meaning it involves two parents, each contributing genetic material. One set of instructions comes from the male in the form of sperm cells, and the other from the female in the form of oocytes, also called egg cells. When these two meet and combine, they create a complete set of genetic information, unique to the new individual. This combination is important. It allows for variation. Every child born from sexual reproduction carries a mix of traits from both parents, but never an exact copy. This diversity is one of nature's most powerful tools. It means that if the environment changes, if the climate shifts, if food sources change, if new challenges arise, some individuals will have traits that help them adapt. Those traits can then be passed on. Reproduction, then, is not just about creating life, but about shaping it for the future. Over many generations, these small variations accumulate, guiding the course of a species' evolution. This is why biologists often say that reproduction is the foundation of both survival and change in living systems. In the human body, the reproductive systems do not act alone to achieve this purpose. They depend on cooperation from other systems. The endocrine system, with its carefully timed release of hormones, initiates and controls reproductive cycles. The nervous system communicates signals between brain and body, guiding responses and behaviors related to reproduction. The circulatory system delivers nutrients and oxygen to reproductive tissues, ensuring they remain healthy and capable of fulfilling their roles. While reproduction is universal among living things, its forms and strategies vary widely. In some species, one individual can produce offspring alone, a process called asexual reproduction. This method creates identical copies of the parent, and while it is efficient, it does not create variation in the same way sexual reproduction does. Other species, like humans, rely entirely on two individuals working together biologically, each providing half the genetic material. Humans also differ from many species in that our reproductive systems are deeply tied to cycles and seasons within an individual's life. From birth, the basic structures are present, but they remain largely inactive through childhood. At puberty, hormonal changes awaken these systems, triggering the production of gametes and the development of physical characteristics linked to reproduction. These changes are gradual, often taking years before the systems are fully mature. Once active, the reproductive systems follow patterns in males, a continuous process of sperm production, and in females, a monthly cycle of oocyte development and release. These patterns are influenced by age, health, environment, and even emotional well-being. Over time, as a person ages, the systems begin to slow, eventually reaching a stage where gamete production ceases. In females, this transition is marked by menopause, the natural end of the menstrual cycle. It is important to remember that the purpose of reproduction is not tied to any one individual's choice or circumstance. Many people do not have children, yet their bodies still carry the capacity for reproduction, at least during certain phases of life. The presence of this system is not a directive, but a biological potential, a built-in ability that is part of the design of our species. The act of reproduction itself, in biological terms, begins when a sperm cell and an oocyte meet. But the preparation for that moment is constant, happening quietly in the background of the body's daily life. In the male system, millions of sperm are produced every day, each carrying half the genetic blueprint. In the female system, oocytes are prepared over many years, each one waiting for the hormonal signal that may bring it to maturity. When these gametes meet under the right conditions, they merge into a single cell called a zygote. This tiny structure, smaller than the point of a pin, contains all the instructions needed to build a new human being. From that single cell, division and growth begin, leading over time to the complexity of a newborn child. The story of reproduction is therefore one of both simplicity and complexity. At its heart, it is the joining of two cells, but surrounding that moment is a web of processes, physical, chemical, and hormonal, that must be precisely timed and coordinated. The systems involved must be healthy, the environment must be suitable, and countless steps must occur in the correct order for new life to begin. It is easy to think of reproduction as a forward-looking process, concerned only with the next generation. Yet it is also deeply rooted in the past. The genetic instructions carried in gametes are the result of a long history, shaped by the lives of ancestors stretching back through centuries. When two gametes merge, they bring together not only the traits of the parents, but echoes of countless generations before them. In this way, reproduction becomes more than a biological function. It is a living archive, a way of preserving the story of life while also allowing it to grow and change. Each new generation is both a continuation and a fresh beginning, a bridge between what has been and what will be. As we move forward in this exploration, we will begin to see how the structures of the reproductive systems serve this purpose. We will look closely at the male system and the female system, each with its own pathway, each with its own specialized roles. We will see how these systems are shaped to protect and guide the gametes, and how they work together to create the possibility of life. For now, you can rest with the thought that reproduction, though it may seem like a single act, is actually a continuous and patient process. It is the hum in the background of human biology, always present, always preparing, always carrying forward the quiet continuity of life. 3. Sexual reproduction, the joining of gametes. In the soft architecture of life, there is a single, pivotal moment when two living cells, each carrying half of the blueprint for a human being, meet and unite. This is the joining of gametes, the essence of sexual reproduction. It is the quiet spark at the very beginning of a new life, and yet it is the result of long preparation by both the male and female reproductive systems. In humans, sexual reproduction is built on the principle of contribution from two individuals. The male provides sperm cells, each one small, highly mobile, and streamlined for travel. The female provides oocytes, larger, nutrient-rich cells, prepared to nurture the earliest stages of development. Though different in form and function, these gametes are perfectly matched in purpose. Together, they create a complete set of genetic instructions for a new organism. A gamete, whether sperm or oocyte, contains just 23 chromosomes, half the number found in most of the body's cells. These chromosomes are like volumes in a library, each containing hundreds or thousands of genes, and each gene carrying the code for a specific trait or function. When the two gametes merge, their chromosomes combine, restoring the total to 46. The resulting single cell, called a zygote, now holds the full genetic library needed to guide the growth of a new human. The process begins in separate places, in separate bodies. In the male, sperm are produced continuously in the testes, each one maturing within the coiled passages of the epididymis before traveling through a network of ducts. In the female, oocytes are not made anew each day. Instead, they have been present since before birth, held in a quiet, suspended state within the ovaries. Each month, under the guidance of hormones, one oocyte, sometimes more, moves toward maturity and is released into the fallopian tube. For fertilization to occur, sperm must travel from the male body into the female reproductive tract. This journey begins during sexual intercourse, when semen, containing millions of sperm suspended in nourishing fluid, is released into the vagina. From there, sperm travel through the cervix and into the uterus, their ultimate destination being the fallopian tube, where an oocyte may be waiting. This journey is long and challenging, and most sperm will not reach the oocyte. Along the way, they are aided by both their own energy reserves and by subtle changes in the female reproductive tract that guide and support their movement. The environment of the cervix and uterus shifts in response to hormonal cycles, becoming more favorable to sperm during the time when fertilization is most likely. If a sperm reaches the oocyte, the final and most delicate stage begins. The sperm must penetrate the protective layers surrounding the oocyte, a process that requires both physical effort and chemical signals. Once a single sperm successfully enters, the oocyte responds by altering its surface, preventing any other sperm from entering. This ensures that the resulting zygote has the correct number of chromosomes. Inside this newly formed zygote, the genetic material from the sperm and the oocyte comes together. Two sets of chromosomes align, and for the first time, a unique genetic identity exists, not quite like either parent, but a blend of both. This cell will begin to divide over and over, following the instructions encoded in its DNA, eventually forming the many types of cells needed to create a complete human body. This first cell is small, but it carries a vast potential. The early divisions produce a cluster of cells that will travel down the fallopian tube toward the uterus, a journey that takes several days. Once in the uterus, if the conditions are right, the developing cluster will attach to the thickened lining of the uterine wall, a process called implantation. Here, nourished and protected, it will continue to grow. The joining of gametes is, in one sense, a simple act, the meeting of two cells, but it is also the point where countless biological processes converge. It relies on the production of gametes over time, on the coordinated function of multiple organs, on the precise timing of hormonal cycles, and on the intricate design of each reproductive system. It is the culmination of preparation that may have been underway for many years before the moment itself. In nature, sexual reproduction serves a purpose beyond the creation of a single new life. By blending genetic material from two individuals, it introduces variation into the population. This variation is the raw material for evolution, allowing the species to adapt to changes in the environment over generations. It is why no two individuals, apart from identical twins, are exactly the same. In humans, this joining is also woven into the fabric of relationships, family lines, and cultural traditions. But here, in the quiet space of learning, we focus on its biology, the precise yet gentle processes that bring two cells together and set life in motion. As we move forward, we will look at the origins of these gametes more closely, exploring the structures that produce them and the pathways they travel. We will see how the male and female reproductive systems are shaped for their respective roles, and how each supports the possibility of this joining. For now, imagine the scene at a microscopic level, the meeting of two cells, each carrying half a story, coming together to write the first sentence of a new one. It is a beginning both humble and extraordinary, a reminder that even the most complex forms of life start from something very small, guided by a quiet, persistent rhythm that has been part of the natural world for as long as life has existed. 4. Shared beginnings in embryonic development. Before birth, before any distinction between male and female anatomy is visible, the foundations of the reproductive systems are already quietly forming. In the earliest weeks of embryonic life, the structures that will one day become either testes or ovaries, and the ducts that will guide gametes in adulthood, all begin from the same basic blueprint. This is the shared beginning, a time when the path of development has not yet been decided. Within the first few weeks after fertilization, the embryo is a rapidly dividing, organized collection of cells, already establishing the framework for every organ system. Among these early formations are the gonadal ridges, small swellings on either side of the developing spine. These ridges are not yet specialized. They have the potential to become either male or female gonads, depending on the genetic and hormonal signals they will soon receive. In the same period, two parallel duct systems begin to develop, the Wolffian ducts and the Mullerian ducts. These are paired tubes that run through the early embryo. At this stage, both sets are present in every embryo, regardless of genetic sex. One set will eventually develop into the reproductive structures of one sex, while the other set will diminish and disappear. Which path is taken depends on the instructions written into the chromosomes and the timing of specific hormonal signals. The deciding factor is found in the sex chromosomes, the 23rd pair of chromosomes in the zygote's genetic makeup. If the pair is XX, the embryo will develop female reproductive structures. If the pair is XY, male structures will form. But these chromosomes do not act alone. They carry instructions that guide the formation of key hormones and proteins, which in turn shape the tissues of the developing reproductive system. In embryos with an XY chromosome pattern, a gene on the Y chromosome called SRY, the sex-determining region of the Y, becomes active around the 7th week of development. This gene instructs the gonadal ridges to form testes. Once present, the testes begin to produce testosterone, a hormone that stimulates the growth of the Wolffian ducts into structures such as the epididymis, vas deferens, and seminal vesicles. At the same time, another substance, called Mullerian inhibiting hormone, MIH, causes the Mullerian ducts to regress. In embryos with an XX chromosome pattern, the absence of the SRY gene means the gonadal ridges develop into ovaries. Without high levels of testosterone or MIH, the Mullerian ducts remain, gradually transforming into the fallopian tubes, uterus, and the upper portion of the vagina. The Wolffian ducts, lacking hormonal support, quietly shrink and disappear. These early developmental choices set the stage for the reproductive systems we will explore in greater detail later. But at this stage in the embryo's life, the process is subtle. The structures are tiny, and changes unfold slowly over weeks. The transformation is guided by a careful balance of timing, hormone levels, and genetic instruction. It is important to note that the reproductive system does not grow in isolation. While the gonads and ducts are forming, other systems are developing alongside them. The urinary system, for instance, shares some of the same early tissue origins. This close relationship explains why the urinary and reproductive systems remain physically connected in many ways throughout life, particularly in males where the urethra serves as a passage for both urine and semen. Even the external genitalia, the structures visible outside the body, begin from the same starting point in all embryos. In the early weeks, they consist of a small genital tubercle, paired swellings, and folds of tissue. Under the influence of hormones, these tissues will either elongate and fuse to form the penis and scrotum in males, or remain shorter and develop into the clitoris and labia in females. But in the earliest stages, their appearance is so similar that it is impossible to tell them apart. This shared beginning is a reminder that the differences between male and female reproductive systems emerge gradually, guided by a complex interplay of genes and hormones. The two systems are not entirely unrelated. They are variations on a common plan, adapted for complementary roles in reproduction. By the time an embryo reaches the end of its first trimester, the basic structures of the reproductive system are in place, though they are still immature and will remain so for many years after birth. They will lie dormant through childhood, quietly maintained by the body until the time comes for them to awaken during puberty. In understanding these beginnings, we see that male and female anatomy are not opposing designs, but parallel developments from a shared origin. This perspective helps explain why certain structures are called homologous, meaning they arise from the same embryonic tissue, but take on different forms and functions in adulthood. For example, the testes and ovaries are homologous, as are the penis and clitoris, and the scrotum and labia majora. As we move forward, we will explore these differences and similarities in greater detail, beginning with a gentle overview of both systems as they appear in the adult body. But for now, picture the quiet unfolding of the earliest stages: two tiny ducts, a pair of ridges, and a small set of folds and swellings, all holding the potential to take one of two paths. It is the first chapter of the reproductive story, written in the language of development long before birth. 5. Overview of male and female systems. By the time we reach adulthood, the two human reproductive systems, male and female, have grown into distinct yet complementary arrangements. Each has its own collection of organs, tissues, and pathways, shaped for its role in the creation of new life. When we step back and look at them as complete systems, we can see them as two halves of a shared biological design, working toward the same goal through different means. The male reproductive system is built around the continuous production and delivery of sperm cells. It extends both inside and outside the body, with structures that protect, nourish, and transport gametes. This system must not only make these cells, but also keep them at the right temperature, supply them with energy, and deliver them to the female reproductive tract. To do this, the male system combines several kinds of organs: the gonads, which produce sperm and hormones, a series of ducts that store and move sperm, and accessory glands that produce the fluid known as semen. Together, These parts form a connected pathway, starting in the testes and ending at the urethral opening at the tip of the penis. The female reproductive system is arranged differently. All of its primary structures are located within the pelvic cavity, protected by the bones and soft tissues of the lower abdomen. Its central function is to produce oocytes and provide a nurturing environment for a potential pregnancy. This means the system must have a safe place for fertilization to occur, a chamber for an embryo to implant and grow, and the ability to deliver a child when the time comes. To meet these needs, the female system includes the gonads, the ovaries, along with paired fallopian tubes, a muscular uterus, and the birth canal, which is the vagina. Surrounding and protecting the opening of the vagina are the external structures collectively called the vulva. Both systems share some structural principles. Each begins with a pair of gonads, testes in the male, ovaries in the female, which are the sources of gametes and sex hormones. From the gonads, the gametes travel through specific pathways. In the male, a long journey through ducts and out of the body. In the female, a shorter journey from the ovary through the fallopian tube toward the uterus. In both cases, the supporting structures are essential. Without them, the gametes would not be able to survive or reach their destination. Hormones play a guiding role in both systems. In males, testosterone supports sperm production and the development of secondary sexual characteristics, such as facial hair and a deeper voice. In females, a cycle of hormones, including estrogen and progesterone, regulates the monthly preparation of the oocyte, the thickening of the uterine lining, and the timing of ovulation. These hormonal patterns are coordinated by the endocrine system, with the brain's hypothalamus and pituitary gland sending signals that influence the gonads directly. Despite their differences, the two systems are intricately matched. The male system's focus is on producing many small, motile gametes, optimized for travel. The female system produces fewer, larger gametes, each one containing the nutrients needed to support the earliest stages of development. The meeting point of these two strategies is fertilization, when the mobility of the sperm meets the readiness of the oocyte. In addition to producing gametes, both systems play roles in sexual function, and in many cases, in bonding between partners. Physical structures, sensory nerves, and blood vessels all work together to create responses that are part of the reproductive process, even if reproduction is not the immediate goal. From a broader perspective, both systems are also connected to the rest of the body in practical ways. The male reproductive tract shares pathways with the urinary system, while the female reproductive system is closely tied to changes in overall physiology during pregnancy. During these times, the circulatory, respiratory, and endocrine systems adjust to meet new demands, showing once again that the reproductive systems do not operate in isolation. When viewed side by side, the male and female reproductive systems reveal a story of complement and cooperation. They are different not in opposition, but in purpose, two sets of structures, each complete in itself, yet designed to work together when conditions align. In the next sections, we will explore these systems in greater detail, beginning with a closer look at the male reproductive pathway, from its external structures to its deepest internal components. But before we step into the details, it is worth holding the full picture in mind. Two systems, two designs, and one shared purpose: the continuation of human life through the meeting of gametes. 6. The male reproductive pathway, an outside and inside tour. The male reproductive system begins with structures you can see and continues deep into the body with a network of organs, ducts, and glands. Together, they form a continuous pathway, guiding sperm from the moment they are produced to the point where they leave the body. It is a design that balances protection, mobility, and efficiency, each part contributing to the success of the whole. The journey starts at the testes, the male gonads. These are oval-shaped organs, usually about 4 to 5 centimeters in length. housed in a loose sac of skin called the scrotum. The scrotum is more than a simple covering. It is a temperature control system. Sperm develop best at a temperature slightly cooler than the body's core. To maintain this, muscles in the scrotum gently adjust the position of the testes, drawing them closer to the body in cold conditions and allowing them to hang farther away in warmth. Inside each testis, tightly coiled tubes called seminiferous tubules are the sites of sperm production. Lining these tubules are specialized cells that divide and mature in a steady rhythm, ensuring a constant supply of new sperm. Once sperm are formed, they are not yet ready for their role. They move into the epididymis, a long, coiled duct lying along the back of each testis. Here, sperm spend time maturing, developing the ability to swim and to recognize and bind with an oocyte. This maturation can take several weeks, during which the epididymis also serves as a storage site until the sperm are needed. When ejaculation occurs, muscular contractions move sperm from the epididymis into the vas deferens, a muscular tube about 30 centimeters long. The vas deferens travels upward into the pelvic cavity, looping over the bladder. This tube is well adapted for transport. Its muscular walls create wave-like movements, known as peristalsis, that help propel the sperm forward. Along the path, the sperm encounter the first of several accessory glands, beginning with the seminal vesicles. These paired glands lie just behind the bladder and produce a thick, alkaline fluid rich in fructose, which serves as an energy source for sperm. This fluid also contains substances that help sperm survive in the acidic environment of the female reproductive tract. The secretions of the seminal vesicles account for the majority of semen's volume. Next, the sperm pass through the ejaculatory ducts, short tubes formed where the vas deferens joins the seminal vesicle ducts. These ducts lead directly into the prostate gland, a firm, walnut-sized organ located just below the bladder. The prostate adds its own fluid to the semen, slightly milky and also alkaline, which helps to further protect and nourish the sperm. It also contains enzymes that keep semen in a liquid state after ejaculation, aiding sperm movement. Just before entering the urethra, sperm receive another contribution from the bulbourethral glands, also known as Cowper's glands. These small, pea-sized glands produce a clear, slippery fluid released prior to ejaculation. This fluid lubricates the urethra and helps neutralize any acidic traces of urine, creating a safer passage for sperm. From here, the combined fluid sperm from the testes and secretions from the accessory glands enter the urethra, the final segment of the pathway. The urethra runs through the length of the penis, a structure made primarily of spongy erectile tissue. This tissue can fill with blood during arousal, creating the firmness needed for penetration. The urethra serves a dual role in males, carrying both urine and semen. Though never at the same time, a small muscular valve ensures that these two functions remain separate. Finally, during ejaculation, strong rhythmic contractions expel semen from the urethral opening at the tip of the penis. The entire pathway, from the testes to the outside world, is a carefully coordinated route designed to protect and deliver sperm efficiently. It is worth noting that each part of this pathway depends on the others. Without the cooling system of the scrotum, sperm would not develop properly. Without the epididymis, they would not mature. Without the accessory glands, they would lack the protective fluids that help them survive the journey ahead. This interdependence mirrors the broader truth of human anatomy. No organ works in isolation. In the next section, we will look more closely at the testes themselves, the central factories of the male reproductive system, and explore how they create both sperm and hormones. This will take us deeper into the fine details of sperm development and the role of testosterone in male physiology. 7. Testis, factories of sperm and hormones. At the very center of the male reproductive system are the testes, a pair of oval organs, each small enough to rest comfortably in the palm of your hand, yet complex enough to sustain one of life's most essential processes. Their dual purpose is simple in statement but intricate in execution: to produce sperm and to secrete the primary male sex hormone, testosterone. Each testis is housed within the scrotum, a loose, protective sac of skin that hangs outside the main body cavity. This position is no accident. Sperm development, known as spermatogenesis, requires a temperature slightly lower than the core body temperature, about 2 to 3 degrees Celsius cooler. The scrotum, along with small muscles like the cremaster muscle and the dartos muscle, adjusts the testes position in response to temperature changes. In cooler weather, the muscles draw the testes closer to the body for warmth. In heat, they relax, allowing the testes to hang farther away, where air can circulate. Beneath the tough outer covering of each testis, called the tunica albuginea, lie hundreds of tightly coiled tubes known as seminiferous tubules. These are the true factories of sperm production. Each tubule is lined with specialized cells that progress through the stages of sperm development. The process begins with spermatogonia, stem-like cells capable of dividing to produce more sperm precursors. Through a series of steps involving cell division and maturation, these cells eventually become fully formed spermatozoa. This transformation is not instantaneous. It takes roughly 64 to 72 days for a sperm cell to develop from its earliest form to full maturity. Along the way, the developing cells are supported by Sertoli cells, sometimes called nurse cells. These cells provide nourishment, structural support, and chemical signals that guide the process. Sertoli cells also form the blood-testis barrier, a protective separation that prevents immune system cells from mistakenly attacking developing sperm, which are genetically unique compared to the rest of the body's cells. Interwoven between the seminiferous tubules are clusters of Leydig cells. These are the hormone-producing cells of the testes, responsible for synthesizing testosterone. This hormone is essential not only for sperm production but also for the development and maintenance of male secondary sexual characteristics, such as facial hair, a deeper voice, and increased muscle mass. Testosterone also influences mood, energy levels, and aspects of sexual behavior. The production of testosterone in sperm is carefully regulated by the endocrine system through a feedback loop involving the brain. The hypothalamus, a small but crucial region in the brain, releases gonadotropin-releasing hormone, GnRH, which signals the pituitary gland to secrete two hormones, luteinizing hormone, LH, and follicle-stimulating hormone, FSH. LH stimulates the Leydig cells to produce testosterone, while FSH works with testosterone to promote spermatogenesis within the seminiferous tubules. Once sperm are fully formed, they are released into the lumen, the central cavity of the seminiferous tubules. From here, they are gently carried along by fluid secretions and tiny hair-like projections called cilia into the rete testis, a network of small channels. They then pass into the efferent ductules and onto the epididymis, where they will continue to mature. The structure of each sperm reflects its purpose. The head contains the nucleus, where the genetic material is stored, and is capped by the acrosome, a small sac filled with enzymes needed to penetrate the outer layers of the oocyte. The midpiece is packed with mitochondria, the cell's energy generators, which fuel the movement of the tail. The tail, or flagellum, provides the propulsion that will help the sperm navigate through the female reproductive tract. The testes work continuously, producing millions of sperm every day, though not all will ever be used in fertilization. This ongoing production is a hallmark of the male reproductive system, ensuring that viable gametes are available whenever needed. Beyond reproduction, the role of the testes in hormone production has far-reaching effects throughout the body. Testosterone influences bone density, red blood cell production, and metabolism. It supports overall vitality, and its decline with age can affect physical strength, mood, and sexual function. In their modest size and unassuming location, the testes hold a vital place in the continuity of life. They are both the starting point of the sperm's long journey and a key source of the hormones that shape male physiology. In the next section, we will follow the sperm as they leave the testes and enter the epididymis, a coiled passageway, where they gain the final skills needed for their journey ahead. 8. Maturation in the epididymis. When sperm leave the seminiferous tubules of the testes, they are physically complete, but not yet truly ready for their task. They have their characteristic shape, a rounded head, a midpiece packed with mitochondria, and a long, slender tail. Yet these structures are still not fully functional. At this stage, the sperm cannot swim effectively, and they cannot successfully fertilize an oocyte. Their next destination, the epididymis, is where they will acquire these essential abilities. The epididymis is a remarkable structure, a single, highly coiled tube that rests along the back surface of each testis. In appearance, it may seem small, but if uncoiled, each one would stretch out to several meters in length. This length is important because it provides time and space for sperm to mature as they slowly make their way from the point where they enter to the place where they will be stored. The epididymis is divided into 3 main regions: the head, caput, the body, corpus, and the tail, cauda. Sperm enter at the head, arriving from the efferent ductules that connect to the rete testis. At this point, they are still largely immobile and lack the specialized surface proteins required for fertilization. In the head region, epithelial cells lining the epididymal walls begin to secrete a carefully balanced fluid mixture. These secretions are rich in glycoproteins, enzymes, and small molecules that interact with the sperm's membrane. Through these interactions, the sperm begin to undergo subtle chemical changes. Their cell membranes become more stable, their internal structures more organized, and their surface proteins start to form into the patterns that will later be crucial for recognizing and binding to an oocyte. As the sperm travel into the body of the epididymis, they encounter an environment designed to encourage gradual functional activation. Here, the composition of the fluid changes, becoming more acidic and containing different proteins and ions. This acidity keeps sperm metabolism low, conserving their energy for the demanding journey they will eventually make through the female reproductive tract. Low metabolic activity is essential because sperm do not replenish their energy stores. Once they leave the male body, they must make do with what they carry in their midpieces. During this stage, sperm also begin to acquire motility. In the testes, their tails are present but largely inactive. Within the epididymal body, chemical signals stimulate changes in the tail's internal axoneme, a structure made of microtubules and motor proteins, such as dynein. These microscopic components start to function in a coordinated way, producing the rhythmic, whip-like movements that will later propel the sperm forward. This is not yet full swimming power, but it is the foundation of motility. The journey through the epididymis is slow, often taking several days, and this gradual pace is part of the design. Rushing this process would result in sperm that are structurally intact but functionally incomplete. Instead, the epididymis acts as a patient tutor, allowing the sperm to develop the endurance, responsiveness, and precision they will need when faced with the challenges of reaching and fertilizing an oocyte. By the time sperm reach the tail region, they are mature in structure and function. Here, they are stored in high concentrations, suspended in a fluid that keeps them dormant but viable. The tail acts as a storage reservoir, holding sperm until ejaculation. If sperm are not used after a certain period, they are broken down and reabsorbed by the body, a natural recycling process that prevents the accumulation of aging or damaged cells. Another remarkable feature of the epididymis is its ability to protect sperm from the immune system. Since sperm carry only half the genetic material of the body's cells, they are considered foreign by the immune system. The blood-testis barrier in the testes already provides significant protection, But the epididymis continues this safeguard. Its epithelial cells and surrounding tissue create a controlled environment where immune cells cannot easily attack or damage the sperm. The relationship between the epididymis and the sperm it houses is both protective and preparatory. It is here that sperm gain capacitation, readiness, the potential to undergo capacitation, a final maturation step that happens later in the female reproductive tract. Capacitation involves changes to the sperm membrane that make it capable of penetrating the oocytes' outer layers. Without the epididymis's preliminary work, capacitation would not be possible. From a structural standpoint, the epididymis is supported by smooth muscle layers that become thicker as one moves toward the tail. During ejaculation, these muscles contract in rhythmic waves, pushing the stored sperm into the vas deferens for the next phase of their journey. This muscular action is coordinated with signals from the nervous system, ensuring that sperm release is timed with the contributions from the accessory glands that will form semen. The epididymis is not merely a passive holding area. It is an active, dynamic environment that fine-tunes sperm for success. Without it, sperm leaving the testes would be incapable of effective movement, unable to recognize an oocyte, and unprepared for the conditions they will face in the female reproductive tract. It is, in every sense, a finishing school for gametes, a place where potential is refined into readiness. When we consider the male reproductive system as a whole, the epididymis plays a critical transitional role. It bridges the highly specialized process of sperm formation in the testes with the equally specialized process of sperm delivery through the vas deferens and beyond. In doing so, it ensures that by the time sperm leave the male body, they have been tested, prepared, and preserved in optimal condition. From here, the path leads to the vas deferens, a sturdy, muscular tube that carries sperm on a rapid but controlled route into the pelvic cavity. Along the way, new elements will be added from the seminal vesicles, prostate gland, and bulbourethral glands, transforming the sperm from individual cells into part of a larger, nourishing, and protective fluid, semen. In the next section, we will follow this route, exploring the vas deferens in detail and learning how it serves as both a transport system and a crucial link between sperm storage and the final stages of the male reproductive pathway. 9. Vas deferens and the transport route. From the tail of the epididymis, mature sperm take their next step along the male reproductive pathway, entering a tube known as the vas deferens. This structure is not especially long compared to the epididymis, about 30 to 45 centimeters, but it is a critical connector. It links sperm storage in the scrotum to the internal structures of the pelvic cavity, carrying its passengers with both protection and precision. The vas deferens begins as a narrow continuation of the epididymal tail. Its initial path takes it upward within the spermatic cord, a bundle of structures that includes blood vessels, lymphatic vessels, nerves, and a layer of connective tissue. The spermatic cord extends from the scrotum through the inguinal canal, a short passage in the lower abdominal wall. This arrangement ensures that the testes and epididymis remain connected to the rest of the body's systems, while still positioned outside the abdominal cavity. The wall of the vas deferens is made up of 3 layers of smooth muscle, an inner longitudinal layer, a middle circular layer, and an outer longitudinal layer. This muscle arrangement allows the vas deferens to perform powerful, wave-like contractions known as peristalsis. Unlike the gentle, slow movement that carries sperm through the epididymis, the contractions here are forceful and coordinated, designed to propel sperm rapidly during ejaculation. The inner lining, or mucosa, of the vas deferens is folded to create a larger surface area, although its role in secretion is minor compared to the accessory glands. The mucosa contributes a small amount of fluid that helps keep sperm suspended and moving smoothly. The lining also protects sperm from sudden changes in pH or temperature as they travel from the relatively cool environment of the scrotum to the warmer pelvic cavity. The vas deferens travels upward from the scrotum into the pelvic cavity, looping over the urinary bladder. Near its end, it widens into a region called the ampulla. This expansion serves as a temporary holding area, allowing sperm to be stored for a short time before mixing with the fluids from the accessory glands. It is at this ampulla that the vas deferens meets the duct from the seminal vesicle. Together, these two ducts form the ejaculatory duct, a short passage that travels through the prostate gland and into the urethra. This point marks a transition from sperm transport to semen transport, as the seminal vesicles contribute their distinctive fluid to the mix. The design of the vas deferens reflects its dual roles. It must safeguard sperm during the quiet, inactive periods between ejaculations, and it must serve as a rapid transit system when the time for release comes. For most of its length, the vas deferens holds sperm in a dormant state, its environment kept slightly acidic to slow metabolism and conserve energy. But when ejaculation is triggered, the muscles in its walls respond almost instantly, pushing sperm forward at considerable speed. This responsiveness is controlled by the autonomic nervous system, specifically the sympathetic division. When sexual stimulation reaches a certain threshold, nerve signals trigger contraction of the smooth muscle layers. These contractions work in concert with similar activity in the epididymis, ejaculatory ducts, and accessory glands to move semen toward the urethral opening. Because the vas deferens is so important in sperm transport, it is also the focus of certain surgical procedures. One example is a vasectomy, a common form of male contraception. In this procedure, each vas deferens is cut and sealed, preventing sperm from entering the semen. The rest of the reproductive system remains functional fluids. From the accessory glands are still produced, but the semen no longer contains sperm. This change has no effect on testosterone production or other hormonal functions, since those are controlled by the testes and endocrine system. Although the vas deferens has a relatively straightforward path compared to some other reproductive structures, its role cannot be understated. Without it, sperm could not move from their storage site in the scrotum to the urethra, and fertilization would be impossible. Its sturdy, muscular design ensures that sperm are delivered intact, protected from physical stress, and timed perfectly with the release of accessory gland fluids. From this point forward in the pathway, sperm will no longer travel alone. At the meeting of the vas deferens and seminal vesicle, they will join with a rich, nourishing fluid that not only sustains them, but also plays a role in helping them survive the journey through the female reproductive tract. This transformation from individual gametes to semen marks the beginning of the final stages of preparation before ejaculation. In the next part, we will explore the seminal vesicles in detail. These paired glands contribute the largest portion of the fluid that makes up semen, and their secretions are tailored to meet the sperm's needs in terms of nutrition, protection, and mobility. Understanding their role will give us a clearer picture of how sperm are equipped for the demanding journey that lies ahead. 10. Seminal vesicles, nourishment and support. At the point where the vas deferens widens into the ampulla and prepares to join the ejaculatory duct, the sperm meet one of their most important partners in the journey toward fertilization, the seminal vesicles. These two elongated, pouch-like glands lie side by side, positioned just behind the urinary bladder and in front of the rectum. Each seminal vesicle is connected to its corresponding vas deferens, and together they form a crucial meeting point where sperm are joined by the largest single portion of the fluid that will become semen. Their role is vital, for they do far more than simply provide liquid. They transform sperm from isolated cells into part of a nutrient-rich, protective medium capable of sustaining life on its journey into the female reproductive tract. Although modest in size, each seminal vesicle is internally complex. Its walls are folded and coiled, creating a large internal surface area for secretion without taking up excessive space within the pelvis. This folding is a hallmark of efficient biological design, enabling the gland to produce a substantial volume of fluid relative to its size. That fluid is not random in composition but carefully tailored to meet the needs of sperm at the moment they leave the male body. The most abundant ingredient is fructose, a simple sugar that serves as the primary energy source for sperm cells. Since sperm do not replenish their energy reserves after ejaculation, this fructose is their fuel supply for the demanding journey ahead. Without it, their ability to move and to reach the oocyte would be sharply reduced. The fluid is alkaline in nature, a property that serves as a counterbalance to the naturally acidic environment of the vagina. This acidity plays an important role in defending the female reproductive tract against infections, but it is hostile to sperm. By raising the pH of semen, the seminal vesicle fluid helps protect sperm from damage, extending their lifespan and improving their mobility once inside the female tract. Alongside fructose and alkalinity, the fluid contains prostaglandins, hormone-like compounds that have subtle but important effects. In the female reproductive tract, prostaglandins can encourage gentle contractions of smooth muscle in the uterus and fallopian tubes, potentially helping to move sperm closer to the site of fertilization. This is an example of how male and female physiology work in quiet coordination, even before the two systems meet in fertilization. Another feature of seminal vesicle fluid is its role in temporarily thickening semen after ejaculation. This is achieved through clotting proteins that cause the semen to coagulate into a gel-like consistency within moments of entering the vagina. This initial thickening is thought to help semen remain close to the cervix, rather than dispersing too quickly. Later, enzymes from the prostate gland will liquefy the semen again, freeing the sperm to begin swimming toward the oocyte. The timing of these changes is carefully balanced. A brief period of stillness, followed by a release into movement. The contribution of the seminal vesicles to semen is not small. In fact, their fluid accounts for about 60 to 70% of the total volume. This means that most of what leaves the male body during ejaculation is produced by these glands rather than by the testes themselves. The sperm, although small in volume, are the essential cargo, while the seminal vesicle fluid is the vessel that carries and protects them. This separation of roles allows each part of the male reproductive system to specialize. The testes focus on producing and maturing sperm, while the seminal vesicles specialize in creating the ideal environment for those sperm to survive and function. The release of fluid from the seminal vesicles is carefully timed. These glands do not simply trickle their secretions into the reproductive tract throughout the day. Instead, they store their fluid until the moment of ejaculation. During sexual arousal, signals from the sympathetic nervous system prepare the smooth muscle walls of the seminal vesicles for contraction. When ejaculation begins, these muscles contract forcefully, pushing the stored fluid into the ejaculatory ducts, just as sperm arrive from the vas deferens. The mixing happens within seconds, ensuring that sperm are bathed in their protective and nourishing medium only when they are about to leave the body. This timing also prevents waste. If seminal vesicle fluid were mixed with sperm too early, it could lose some of its chemical properties before reaching the female tract. By keeping sperm and seminal vesicle fluid separate until the final stage, the male reproductive system preserves the freshness and potency of both. This efficiency is a recurring theme in reproductive anatomy, where resources are conserved and used with precision. The importance of the seminal vesicles is underscored when we consider what would happen without them. Without their fluid, sperm would travel in a much smaller volume of liquid, with little protection from the acidic vaginal environment. They would have no immediate energy supply in the form of fructose, and no prostaglandins to assist their progress in the female tract. Fertilization would still be possible, but the chances of sperm surviving long enough to reach the oocyte would be greatly reduced. In evolutionary terms, such a disadvantage would have serious implications for reproductive success. It is also worth noting that the seminal vesicle's role is not unique to humans. Many mammals have similar glands that produce alkaline, nutrient-rich fluids for sperm. The composition of these fluids varies between species, depending on differences in reproductive biology, but the principle remains the same. Gametes are given additional resources to improve their chances of success. This suggests that the adaptation of accessory gland secretions is a deeply rooted feature of reproductive evolution. When viewed within the broader context of the male reproductive system, the seminal vesicles are an example of quiet but powerful support. They work in the background, building and storing their contribution without fanfare, ready to release it at precisely the right moment. Their role is not in producing life directly, but in ensuring that the cells which carry the potential for life are given the best possible start. By providing nourishment, protection, and chemical cues, they help sperm overcome the many obstacles between ejaculation and fertilization. From here, the combined contents of the vas deferens and seminal vesicles enter the ejaculatory ducts, passing through the prostate gland. This next stage will add another layer of preparation, as prostatic fluid contributes enzymes, additional alkalinity, and compounds that help semen change its consistency in ways that benefit sperm movement. Together, the secretions from the seminal vesicles and prostate create a fluid environment that is as carefully crafted as any natural habitat, one that balances protection, nourishment, and mobility in service of a single purpose: the continuation of life. 11. The prostate gland, enzymes, and protection. As semen continues its journey through the male reproductive pathway, it passes through an organ that adds a final layer of refinement before it is ready to leave the body: the prostate gland. This single, walnut-sized gland lies just below the urinary bladder, encircling the urethra like a ring. Its central position means that it is traversed by both the urinary and reproductive pathways, a design that requires careful control to ensure that urine and semen never travel together at the same time. The prostate's role is subtle yet essential. It produces a fluid that enhances sperm's mobility, stability, and overall chances of surviving the long journey through the female reproductive tract. This contribution, though modest in volume compared to that of the seminal vesicles, has a disproportionate impact on the final quality of semen. The prostate gland's location and structure are tailored to its function. Shaped somewhat like an inverted cone, its base lies against the bladder, while its apex points downward toward the urogenital diaphragm. Internally, the gland is composed of clusters of secretory cells, surrounded by a supportive framework of smooth muscle and connective tissue. These glandular units open into small ducts that empty directly into the prostatic portion of the urethra. During ejaculation, the smooth muscle within the prostate contracts, pushing its fluid through these ducts at precisely the same time that sperm and seminal vesicle fluid enter the urethra via the ejaculatory ducts. This careful coordination ensures that all components mix evenly, creating a uniform semen mixture. The fluid from the prostate gland is slightly acidic to neutral in pH and accounts for about 20 to 30% of semen's total volume. It contains a range of substances that directly benefit sperm. Among the most important are enzymes such as prostate-specific antigen, PSA, which plays a role in liquefying semen after it has been temporarily thickened by proteins from the seminal vesicles. This liquefaction is vital. While the initial coagulation helps semen remain in place within the vagina, it would be counterproductive to leave sperm trapped in a gel-like matrix. By breaking down this matrix after about 15 to 30 minutes, PSA releases the sperm, allowing them to begin swimming toward the cervix. Without this timed liquefaction, sperm mobility would be greatly reduced, and the chance of successful fertilization would decline. The prostate's secretions also include citric acid, which may play a role in maintaining the correct ionic environment for sperm metabolism, as well as zinc, an important mineral for stabilizing sperm cell membranes and genetic material. Additionally, the fluid contains small amounts of enzymes that help protect sperm from oxidative damage, the chemical wear and tear that can occur in the reactive environments they encounter. These protective measures are essential because sperm, though structurally specialized, are also delicate. Their membranes are easily damaged, and their genetic cargo must remain intact for fertilization to succeed. The prostate's contribution helps ensure that sperm remain viable during the critical window after ejaculation. The muscular tissue of the prostate not only aids in delivering its own fluid, but also serves as part of the pumping mechanism that propels semen through the urethra during ejaculation. This muscular action, coordinated with contractions in the vas deferens, seminal vesicles, and pelvic floor muscles, creates the force that expels semen from the body. In this way, the prostate's role is both chemical and mechanical. It supplies important components of the semen while also assisting in its delivery. From a broader perspective, the cooperation between the prostate and other accessory glands is an example of precise biological teamwork. The seminal vesicles provide the main nutrient supply and create the initial structure of semen. The prostate modifies that structure at just the right time, ensuring that sperm can move freely when it matters most. The bulbourethral glands, which act earlier in the process, prepare the urethra itself with a lubricating, pH-neutralizing secretion so that semen passes through without encountering harmful residues. Together, these glands create a supportive environment for sperm that begins within the male body and continues into the female reproductive tract. If the prostate were removed from this sequence, semen would lack key enzymes needed for timely liquefaction, as well as some of the minerals and protective compounds that help sperm survive. Fertility would be affected, not necessarily because sperm could not leave the body, but because their quality, mobility, and longevity would be compromised. This is why prostate health is closely linked to reproductive health, even though the prostate does not produce sperm itself. The passage of semen through the prostate is brief, yet during this time it undergoes a transformation from a nutrient-rich but static fluid into a carefully balanced medium, primed for both stability and movement. The timing of this process is as important as its chemistry. Semen must remain intact long enough to reach the cervix, but must also become free-flowing in time for sperm to move toward the oocyte. The prostate's contributions strike this balance, completing the preparation that began in the testes and epididymis and was expanded in the seminal vesicles. From here, semen enters the final segment of the male reproductive pathway, the urethra. This shared passageway for urine and semen will be used exclusively for reproductive purposes during ejaculation, as reflexes prevent the simultaneous flow of urine. The semen will also receive its final, small addition from the bulbourethral glands, which provide lubrication and pH adjustment for the urethral lining. Once these final touches are complete, the muscular contractions of the pelvic floor and the rhythmic activity of the smooth muscles will expel semen from the body, beginning its journey into the female reproductive system. In the next section, we will examine the bulbourethral glands in detail. Though small in size, these glands make an important final contribution to the semen and prepare the male urethra for its role in the reproductive process. Their work is quick and often unnoticed, yet it is another example of how even the smallest components of the reproductive system play essential roles in supporting the possibility of new life. 12. The bulbourethral glands. Lubrication and preparation. As semen nears the final stage of its passage through the male reproductive system, it receives a last, subtle contribution from a pair of small but highly specialized glands, known as the bulbourethral glands. Also called Cowper's glands, these two pea-sized structures are tucked away beneath the prostate gland, nestled within the urogenital diaphragm at the base of the penis. Though they are modest in size and often overlooked in general discussions of reproduction, their role is both strategic and essential, providing a crucial preparation step that takes place even before ejaculation begins in full. The bulbourethral glands are connected to the urethra by short ducts, each only a few centimeters in length. During sexual arousal, long before semen is released, these glands respond to signals from the nervous system by secreting a clear, slippery fluid that enters the urethra. This secretion, sometimes referred to as pre-ejaculate, performs two main functions: lubrication and neutralization of acidity. The urethra, in its everyday role as part of the urinary system, often contains residual traces of urine. Urine is slightly acidic, and its chemical residues could harm sperm if they were to pass through unprotected. The fluid from the bulbourethral glands gently washes along the urethral lining, reducing acidity and creating a safer environment for the semen that will soon follow. Lubrication is the second vital role of this pre-ejaculatory fluid. As semen passes through the urethra during ejaculation, the smooth flow of its movement is aided by this slippery layer, reducing friction and protecting the integrity of the sperm cells. This lubrication also serves a role in intercourse itself, easing the passage of the penis into the vagina and thereby supporting the transfer of semen. While the amount of fluid produced by the bulbourethral glands is small compared to the contributions of the seminal vesicles and prostate, its timing makes it disproportionately important. The secretion occurs early, before the release of sperm or the main volume of seminal fluid, ensuring that the urethra is already prepared when ejaculation begins. The composition of bulbourethral gland fluid is simple but effective. It is mostly water, providing the necessary liquid base for lubrication, but it also contains mucus and small quantities of alkaline compounds. These alkaline substances help counter any remaining acidity in the urethra after the initial wash of fluid. The mucus gives the secretion a smooth, cohesive texture, allowing it to coat the urethral lining evenly, rather than simply flowing past in a thin stream. This coating effect ensures that the urethra remains protected throughout ejaculation. From a mechanical perspective, the bulbourethral glands operate with precision. Their ducts open directly into the urethra, so the moment their smooth muscle fibers contract under sympathetic nervous control, the fluid enters the passageway and begins its work. This activity is part of a larger sequence of events that unfold in rapid coordination during sexual arousal and climax, involving the testes, epididymis, vas deferens, seminal vesicles, prostate, and the muscular structures of the pelvic floor. One way to understand the significance of these glands is to imagine the journey of sperm without their contribution. Without the protective alkaline layer, sperm would pass directly through a urethra, potentially lined with traces of acidic urine, risking damage to their delicate membranes. Without the lubrication, semen would still reach the outside of the body, but the increased friction might alter the consistency of the ejaculate or affect the smoothness of its delivery. The bulbourethral glands thus play the role of a careful host, preparing the final stretch of the pathway so that the guests, the sperm, and their surrounding seminal fluid can move through unhindered and undamaged. In evolutionary terms, the development of bulbourethral glands in humans and other mammals represents another example of fine-tuning in reproductive anatomy. The reproductive process is filled with potential obstacles: changes in pH, variations in temperature, structural barriers, and time constraints. The gland's early intervention is a safeguard against one of these challenges, ensuring that a potentially hostile segment of the reproductive pathway is rendered safe before the sperm even arrive there. Their contribution is quiet, almost invisible, yet it addresses a vulnerability that could otherwise reduce the chances of successful fertilization. The position of the bulbourethral glands in the sequence of events is also a reminder of how carefully timed the entire reproductive process is. Each structure in the male system activates in the right order, its output blending seamlessly with what came before and what comes after. The bulbourethral glands act before the arrival of semen. The seminal vesicles and prostate contribute during ejaculation, and the muscular contractions of the vas deferens and pelvic floor coordinate the final expulsion. In this way, every part of the system works in harmony, with no wasted steps. Once their task is complete and the urethra is prepared, the bulbourethral glands have no direct role in the remaining passage of semen. However, Their early action is essential for the quality of the final product. By the time semen enters the urethra, a mixture of sperm from the testes and epididymis, nutrient-rich fluid from the seminal vesicles, and enzymatic contributions from the prostate. It does so in an environment already made favorable for its survival and smooth movement. This means that when the rhythmic contractions of the pelvic floor muscles and reproductive ducts begin, there is no need for the body to pause or make adjustments. The path is ready, and the fluid flows unimpeded toward the outside world. From here, the journey of semen passes into its final stage within the male body, traveling the length of the urethra and exiting through the opening at the tip of the penis. In this last segment, the reproductive pathway overlaps entirely with the urinary pathway, a dual role made possible by a system of valves that ensure the two functions do not interfere with one another. During ejaculation, the passage from the bladder is closed, so no urine can mix with semen, and all muscular effort is directed toward propelling the semen outward. It is this final phase, the act of ejaculation itself, that we will explore next. Following semen from its fully prepared state within the urethra to the moment it begins its separate journey within the female reproductive system. 13. Ejaculation, the final stage of the male pathway. Ejaculation is the culmination of a long sequence of coordinated events within the male reproductive system, a point at which all of the preparatory work done by the testes, epididymis, vas deferens, and accessory glands comes together in a single, synchronized release. By the time semen reaches this stage, every component has already been carefully prepared. The sperm have been matured and stored. The seminal vesicles have infused them with nutrients and protective alkalinity. The prostate has added enzymes to manage their environment, and the bulbourethral glands have primed the urethra with lubrication and pH balance. Ejaculation is the final act of delivery, moving semen from inside the male body into the external environment and potentially into the female reproductive tract where fertilization may occur. This process is not a single action but rather a rapid sequence of muscular and neural events, traditionally divided into two main phases: emission and expulsion. Emission is the first phase, during which the various components of semen are moved into position within the urethra. Smooth muscle contractions within the walls of the vas deferens begin by propelling stored sperm from the ampulla toward the ejaculatory ducts. At the same time, the seminal vesicles contract, releasing their nutrient-rich, alkaline fluid into the mix, followed by the prostate gland, adding its enzymatic and stabilizing secretions. These contributions blend in the prostatic urethra, forming a complete semen mixture. The autonomic nervous system, particularly the sympathetic branch, governs this phase, ensuring that each gland and duct contracts at precisely the right moment so that all components arrive together. The second phase, expulsion, is the actual release of semen from the body. This is driven by rhythmic contractions of skeletal muscles in the pelvic floor, particularly the bulbospongiosus and ischiocavernosus muscles, along with continued activity from the smooth muscle in the urethra and reproductive ducts. These contractions occur in rapid succession, creating pulses of pressure that propel semen along the length of the urethra and out through the external opening at the tip of the penis. The sequence is so precisely timed that each contraction sends a small volume of semen forward, ensuring a steady flow rather than a single burst. This wave-like release is advantageous, as it allows semen to be deposited effectively within the female reproductive tract, sometimes closer to the cervix, increasing the chances that sperm will successfully begin their upward journey. Throughout ejaculation, the urinary pathway is temporarily closed off by the internal urethral sphincter, a ring of smooth muscle located at the junction of the bladder and urethra. This ensures that urine cannot enter the urethra and mix with semen, protecting sperm from the harmful acidity and osmotic differences of urine. This closure also prevents semen from traveling backward into the bladder, a condition known as retrograde ejaculation, which, while not harmful in itself, can interfere with fertility. Ejaculation is also accompanied by the culmination of sexual climax, or orgasm, in the male. While orgasm and ejaculation are closely linked, they are not identical. Orgasm is the sensory and psychological experience associated with climax, while ejaculation is the physical act of releasing semen. In most cases, the two occur together, driven by the same coordinated nervous system activity, but they can occur separately in certain circumstances. From a reproductive standpoint, ejaculation is the functional endpoint of the male role in natural conception, delivering semen into a location where sperm have the best possible chance of encountering and fertilizing an oocyte. The volume of semen released during ejaculation varies, but is typically between 2 and 5 milliliters, with each milliliter containing tens of millions of sperm. Despite these numbers, only a fraction of the sperm will survive the conditions of the female reproductive tract, and fewer still will reach the site of fertilization. This reality underscores the importance of every stage that precedes ejaculation. Each gland and organ in the male reproductive system has contributed to maximizing the survival and functionality of sperm before they even leave the body. After ejaculation, the body begins a recovery period known as the refractory phase. During this time, physical and neural mechanisms prevent immediate repetition of the process. Muscles relax, blood flow to erectile tissues decreases, and the glands return to their resting state, ready to slowly rebuild their secretions. The length of the refractory phase varies greatly between individuals and is influenced by factors such as age, overall health, and hormonal balance. From a reproductive perspective, this period allows the body to conserve resources and ensure that future ejaculations can still produce semen of adequate volume and quality. Ejaculation is thus both an end and a beginning. It is the endpoint of the male reproductive pathway, marking the conclusion of sperm's internal journey, from formation in the testes to release into the outside world. At the same time, it is the starting point of the next stage, sperm's passage into the female reproductive system, where entirely new challenges await. Once deposited in the vagina, sperm face a gauntlet of physical and chemical barriers, navigating the acidic environment, passing through the cervical opening, traveling the length of the uterus, and reaching the fallopian tubes where fertilization may take place. The success of this next stage depends heavily on the preparation completed in the male system, and ejaculation is the moment that transitions this preparation into action. From a biological viewpoint, the act of ejaculation reflects the remarkable precision and cooperation between multiple systems: nervous, muscular, endocrine, and reproductive, all working together in less than a few seconds of coordinated activity. The smooth and skeletal muscles contract in harmony, glandular secretions merge in exact proportions, and structural pathways open and close at precisely the right time. This level of integration is a testament to the evolutionary refinement of reproductive processes, where efficiency and timing can make the difference between success and failure in the continuation of a species. In the next part, we will follow semen into the female reproductive tract, tracing the path sperm must take and the conditions they must overcome. While the male system has provided them with every possible advantage, the journey within the female body presents a new set of challenges that will determine whether fertilization ultimately occurs. This exploration will give us a full picture of the reproductive process, linking the preparation within the male body to the complex and selective environment of the female reproductive system. 14. Sperm's journey in the female reproductive system. Once ejaculation has taken place, semen enters the female reproductive tract, beginning the second half of the reproductive journey. This stage is vastly different from the controlled and protective environment of the male body. Here, sperm must navigate a pathway filled with physical barriers, chemical challenges, and directional uncertainty. From the moment they are deposited in the vagina, the clock begins to tick on their survival. Every structural and chemical preparation that took place in the male reproductive system now faces its test. The goal is singular to reach and fertilize an oocyte, but the route is long, and only a small fraction of the millions of sperm released will come close to achieving it. The journey begins in the vagina, a muscular, elastic canal lined with a mucous membrane. While its design allows it to stretch and adapt during intercourse and childbirth, it is also naturally maintained at an acidic pH. This acidity plays a critical role in preventing infections by limiting the growth of harmful bacteria and other microorganisms. However, it also poses a hostile environment for sperm, which are sensitive to pH changes. The alkaline fluid from the seminal vesicles helps neutralize some of this acidity, creating a brief window during which sperm can survive and begin moving toward the cervix. Even with this buffering effect, many sperm are rendered immobile or non-viable in the vaginal environment within minutes. This is the first major point of attrition in the journey. Those that survive proceed to the cervix, the narrow opening at the lower end of the uterus. The cervix is not simply an open doorway. It is a selective gateway that can either aid or hinder sperm passage, depending on timing, within the female reproductive cycle. At most times, the cervical canal is filled with thick mucus that acts as a barrier to sperm, preventing their entry into the uterus. Around the time of ovulation, however, the mucus changes in composition and becomes thinner, clearer, and more alkaline. This fertile cervical mucus forms microscopic channels that guide sperm forward while filtering out those with structural defects or weak motility. In this way, the cervix acts as both a gatekeeper and a quality control checkpoint, ensuring that only the strongest, most viable sperm move deeper into the reproductive tract. Once past the cervix, sperm enter the uterus, a muscular, hollow organ whose walls are lined with the endometrium. This central cavity is considerably larger than the narrow cervical canal, and sperm must rely on both their own motility and the assistance of uterine contractions to move forward. These contractions, influenced in part by prostaglandins from the semen and hormonal signals in the female body, create gentle currents that can carry sperm toward the fallopian tubes. The uterine environment is less hostile than the vagina, but it still presents challenges, including immune cells that may target and destroy sperm as foreign invaders. For many sperm, the uterus becomes the end of the journey, with only a fraction successfully navigating to the next stage. At the upper corners of the uterus, the pathway splits into two fallopian tubes, also known as oviducts. Each tube extends toward an ovary and ends in a fringe of finger-like projections called fimbriae. Sperm entering this region must choose the correct tube, the one containing the oocyte. This choice is largely a matter of chance, though chemical signals released by the egg and surrounding cells may play a role in guiding sperm toward the correct side. The fallopian tubes are narrow, and their inner lining is covered in cilia, tiny, hair-like projections that beat rhythmically to help transport the egg toward the uterus. Sperm must swim against this movement, expending significant energy as they make their way toward the site of fertilization. During their passage through the female tract, sperm undergo a final maturation step known as capacitation. This process involves biochemical changes to the sperm's membrane that increase its ability to bind to and penetrate the oocyte's outer layers. Capacitation also increases the responsiveness of the sperm's tail to calcium, enhancing motility. Interestingly, capacitation does not occur within the male reproductive system. It is triggered by the conditions inside the female tract. Without it, even sperm that reach the egg would be unable to fertilize it. This final transformation ensures that only sperm that have successfully navigated the challenges of the journey are fully capable of achieving fertilization. The site of fertilization is usually the ampulla, a widened portion of the fallopian tube located closer to the ovary than the uterus. Here, sperm encounter the oocyte surrounded by a protective layer of cells called the corona radiata and an outer shell known as the zona pellucida. To penetrate these layers, sperm release enzymes from the acrosome, a specialized, cap-like structure on their heads. This acrosomal reaction allows a single sperm to break through and fuse with the oocyte's plasma membrane. Once this fusion occurs, the egg initiates changes that prevent any additional sperm from entering, ensuring that fertilization results in the correct number of chromosomes. It is remarkable to consider that from the hundreds of millions of sperm released in a single ejaculation, only a few dozen may reach the immediate vicinity of the egg, and typically only one succeeds in fertilizing it. The entire journey, from deposition in the vagina to potential fertilization in the fallopian tube, may take only a matter of hours, though sperm can survive within the female reproductive tract for up to 5 days in favorable conditions. This extended survival window means that fertilization can occur even if intercourse takes place several days before ovulation. From a broader biological perspective, the challenges faced by sperm in the female reproductive system serve as a form of natural selection, ensuring that only the most motile, structurally sound, and well-prepared sperm have the opportunity to contribute their genetic material to the next generation. The entire process, from maturation in the male body to capacitation in the female tract, reflects a finely tuned partnership between two systems, each designed to play its part in the continuation of life. In the next part, we will shift focus to the female reproductive system itself, exploring its structures, functions, and the ways in which it supports not only fertilization, but also the development of new life in the stages that follow. This will complete the full picture of how the male and female systems work together, each with its own complexity and elegance, to make reproduction possible. 15. Overview of the female reproductive system. The female reproductive system is a network of organs, ducts, and supporting structures designed not only to enable fertilization, but also to sustain and nurture developing life until birth. Unlike the male system, which is primarily focused on producing and delivering sperm, the female system must perform multiple roles: producing eggs, receiving sperm, providing the site for fertilization, supporting embryonic and fetal development, and eventually enabling delivery. These tasks require a precise coordination of anatomy, hormonal regulation, and cyclical changes that repeat month after month during the reproductive years. Each component of the system contributes to these functions, forming an interconnected pathway that begins externally and extends deep within the pelvic cavity. The external entry point to the female reproductive system is the vaginal opening, part of the vulva, which includes structures such as the labia majora and labia minora that help protect the internal organs. The vagina itself is a muscular, elastic canal that extends inward, connecting to the cervix at its upper end. Its design allows it to serve multiple purposes: receiving sperm during intercourse, providing a passageway for menstrual flow to leave the body, and forming part of the birth canal during delivery. The lining of the vagina is rich in blood vessels and mucus-producing cells, and its environment is maintained at a naturally acidic pH to discourage infections. This acidity is temporarily moderated during the fertile period, or when semen is introduced, creating a window for sperm survival. Above the vagina lies the cervix, a narrow cylindrical structure that forms the lower end of the uterus. The cervix plays a dual role. It acts as a gateway between the vagina and the uterus and functions as a selective filter for sperm. Its mucus changes in consistency throughout the menstrual cycle, becoming thin and watery around ovulation to facilitate sperm passage, and thickening at other times to create a barrier. The cervix also has the ability to open slightly during ovulation, menstruation, and childbirth, adapting to the different stages of reproductive function. The uterus itself is a hollow, muscular organ shaped like an inverted pear, situated centrally in the pelvic cavity. It has a thick, muscular wall known as the myometrium, and a specialized inner lining called the endometrium. The endometrium is highly responsive to hormonal signals, thickening each month in preparation for a possible pregnancy. If fertilization occurs, the embryo implants into this lining, and the uterus begins the process of supporting its growth. If no fertilization takes place, the thickened lining is shed during menstruation, beginning a new cycle. The uterus is remarkable in its ability to expand during pregnancy, accommodating the developing fetus while maintaining its structural integrity. Extending from the upper corners of the uterus are the fallopian tubes, also called uterine tubes or oviducts. These slender muscular tubes are lined with cilia and specialized secretory cells that help guide an egg from the ovary toward the uterus. At the far end of each tube are the fimbriae, delicate, finger-like projections that sweep over the surface of the ovary during ovulation, capturing the released egg and guiding it into the tube. Fertilization typically occurs within the ampulla, the wider portion of the fallopian tube, and the resulting zygote begins its journey toward the uterus for implantation. The fallopian tubes are not just passive conduits. Their muscular contractions and ciliary action play an active role in moving the egg or early embryo to its next destination. The ovaries, located on either side of the uterus, are the primary reproductive organs in the female system. Each ovary has a dual function, producing and releasing eggs, ova, and secreting hormones such as estrogen and progesterone. At birth, the ovaries contain all the eggs a woman will ever have, each one enclosed in a small structure called a follicle. These eggs remain dormant until puberty, when hormonal signals from the brain initiate the monthly menstrual cycle. During each cycle, several follicles begin to mature, but usually only one reaches full maturity and releases its egg in a process called ovulation. The hormones secreted by the ovaries not only regulate this cycle but also influence the condition of the uterus, the cervical mucus, and other aspects of reproductive readiness. Surrounding and supporting these internal structures are various ligaments and connective tissues that hold the uterus, ovaries, and fallopian tubes in place within the pelvis. These supports allow for slight movement important during pregnancy and childbirth while maintaining overall stability. The female reproductive system is also closely integrated with the circulatory and nervous systems, which provide the blood flow and signaling necessary for its complex functions. What sets the female reproductive system apart is its cyclical nature. Unlike the continuous sperm production in the male system, egg release follows a monthly rhythm, regulated by hormones such as follicle-stimulating hormone, FSH, luteinizing hormone, LH, estrogen, and progesterone. This cycle coordinates the readiness of the ovary, the receptivity of the uterus, and the openness of the cervix. The timing of these changes is critical. An egg remains viable for only about 12 to 24 hours after ovulation, so the entire system works toward aligning sperm arrival with this short window of opportunity. The adaptability of the female reproductive system extends beyond its fertile years. After menopause, when ovulation ceases, the system gradually shifts away from its reproductive functions, though its structures remain part of the body's overall anatomy. Hormonal changes at this stage affect not only the reproductive organs but also other systems, reflecting the interconnected nature of reproductive hormones with overall health. From a biological perspective, the female reproductive system is designed for efficiency and selectivity. Each stage of the reproductive pathway, from egg production to the nurturing of a developing fetus, is finely tuned to optimize the chances of successful conception and healthy development. Every structure, from the microscopic cilia in the fallopian tubes to the muscular walls of the uterus, plays its part in this coordinated process. In the next section, we will explore the menstrual cycle in detail, examining the hormonal rhythms that guide the preparation of the ovary and uterus, the timing of ovulation, and the conditions that make fertilization possible. This deeper look will reveal how the female reproductive system's cyclical changes create recurring windows of opportunity for conception, and how these rhythms are synchronized with the male system's contributions to reproduction. 16. The menstrual cycle, rhythms and phases. The menstrual cycle is the repeating rhythm that governs the activity of the female reproductive system, a carefully timed sequence of hormonal and physical changes that prepares the body for the possibility of pregnancy. Unlike the constant production of sperm in the male system, the female cycle is a monthly process, typically lasting about 28 days. Although variations of several days in either direction are completely normal, each cycle begins with the first day of menstrual bleeding and continues until the day before the next period begins. Within that span, multiple systems, the ovaries, the uterus, and the endocrine network, coordinate in a way that alternates between building and resetting the conditions needed for conception. At the heart of the menstrual cycle are hormones, the chemical messengers that link the brain to the reproductive organs. The hypothalamus, a small structure in the brain, acts as the master timekeeper, releasing gonadotropin-releasing hormone, or GnRH, in pulses. This hormone stimulates the pituitary gland, located just beneath the brain, to release two other hormones, follicle-stimulating hormone, FSH, and luteinizing hormone, LH. These travel through the bloodstream to the ovaries, where they direct the growth and development of follicles, the small sacs in which immature eggs are stored. At the same time, the ovaries respond by producing estrogen and later progesterone, hormones that act on the uterus to prepare it for the potential arrival of a fertilized egg. The menstrual cycle is usually divided into three main phases: the follicular phase, ovulation, and the luteal phase. The follicular phase begins on the first day of menstruation, when the previous cycle's unfertilized egg and the thickened endometrial lining are shed. Menstrual bleeding typically lasts 3 to 7 days, during which the body is essentially clearing the way for a new cycle to begin. FSH levels start to rise, prompting several ovarian follicles to grow. As these follicles develop, they produce increasing amounts of estrogen. This hormone has multiple effects. It encourages further follicle growth, influences the consistency of cervical mucus to make it more sperm-friendly, and stimulates the endometrium in the uterus to begin thickening again. Toward the end of the follicular phase, one follicle usually becomes dominant, outpacing the others in growth and preparing for the release of its egg. Ovulation marks the midpoint of the cycle and is triggered by a surge in LH, a response to the rising estrogen levels produced by the dominant follicle. This surge causes the follicle to rupture, releasing the egg into the pelvic cavity. The fimbriae at the end of the fallopian tube sweep over the ovary to capture the egg, guiding it into the tube where fertilization may occur. Ovulation is the peak of fertility, with the egg remaining viable for only about 12 to 24 hours. Sperm present in the reproductive tract before or shortly after ovulation have the best chance of encountering and fertilizing the egg. Following ovulation, the luteal phase begins. The ruptured follicle transforms into a temporary endocrine structure called the corpus luteum, which secretes progesterone, along with smaller amounts of estrogen. Progesterone's main role is to maintain the thickened endometrium, creating a nutrient-rich, stable environment in case a fertilized egg arrives. It also causes the cervical mucus to thicken again, forming a barrier to sperm and potential pathogens. If fertilization occurs, the developing embryo will produce human chorionic gonadotropin, a hormone that signals the corpus luteum to continue producing progesterone until the placenta takes over this function. If fertilization does not occur, the corpus luteum begins to degenerate after about 2 weeks, leading to a drop in both progesterone and estrogen levels. Without these hormones to sustain it, the endometrial lining breaks down and menstrual bleeding begins, marking the start of a new cycle. This transition underscores the cyclical nature of the system building toward a possible pregnancy, and then resetting if that pregnancy does not happen. The menstrual cycle is not just a biological schedule. It is a finely tuned interplay between different organs and feedback loops. The hypothalamus adjusts NRH release based on estrogen and progesterone levels, creating a dynamic system in which each phase influences the next. The cycle can be influenced by numerous factors, including stress, changes in body weight, illness, and age. During the teenage years, cycles may be irregular as the hormonal system matures. As menopause approaches, cycles often become less predictable before stopping altogether. From a reproductive perspective, the menstrual cycle serves two essential purposes: the maturation and release of an egg, and the preparation of the uterus to receive and support a potential embryo. The follicular phase ensures that an egg is ready. Ovulation provides the opportunity for fertilization, and the luteal phase creates the conditions for implantation. Each phase is distinct but inseparably linked, and even small disruptions in timing or hormone levels can affect fertility. From a broader biological point of view, the menstrual cycle is a remarkable example of recurring readiness, a repeating invitation for new life that continues through decades of reproductive potential. It is a rhythm that reflects both precision and adaptability, capable of functioning consistently under stable conditions, yet flexible enough to respond to the demands of health, environment, and life stage. The cycle's predictability is part of what allows human reproduction to be planned or anticipated, while its inherent variability speaks to the complexity of hormonal regulation. In the next section, we will turn to the process of ovulation in greater detail, exploring not only how an egg is released, but also the changes that occur within the ovary and surrounding structures to make this possible. This closer look will highlight the exact moment when the female system offers its opportunity for fertilization and how this moment is synchronized with the male system's contributions. 17. Ovulation, release, and readiness. Ovulation is the pivotal event of the menstrual cycle, the moment when the ovary releases a mature egg into the fallopian tube, making fertilization possible. This brief window of opportunity is the culmination of days of preparation within the ovary, guided by a precise orchestration of hormonal signals. It represents the transition from potential to availability, when the female reproductive system shifts from preparing an egg to positioning it for union with sperm. While ovulation itself occurs over a matter of minutes, the lead-up and after effects span days, making it one of the most critical and carefully regulated processes in human reproduction. The process begins during the follicular phase of the menstrual cycle, when several ovarian follicles start to mature under the influence of follicle-stimulating hormone, FSH, from the pituitary gland. Each follicle contains an immature egg surrounded by layers of supporting cells. As the follicles grow, they secrete increasing amounts of estrogen, which has multiple roles. It supports continued follicle development, influences the condition of the uterine lining, and modifies cervical mucus to be more receptive to sperm. Among the developing follicles, one usually becomes dominant, a process thought to be driven by its greater sensitivity to FSH and its ability to produce higher levels of estrogen. This dominant follicle outpaces the others, which gradually regress and do not release eggs in that cycle. As estrogen levels from the dominant follicle rise, they eventually reach a threshold that triggers a sudden surge of luteinizing hormone, LH, from the pituitary gland. This LH surge is the direct signal for ovulation. It initiates a cascade of events within the dominant follicle. Enzymes begin breaking down the follicle wall, the fluid inside increases in volume and pressure, and the follicle bulges outward against the surface of the ovary. In the hours following the LH surge, the egg completes its maturation process, progressing from the final stages of meiosis to become a secondary oocyte capable of being fertilized. When the follicle wall finally ruptures, the mature egg is released into the pelvic cavity, near the open end of the fallopian tube. The fimbriae, the delicate finger-like extensions at the tube's opening, are positioned to catch the egg and guide it into the tube. This capture is aided by gentle, rhythmic movements of the fimbriae and by currents in the surrounding fluid, ensuring that the egg does not drift away from the reproductive tract. Once inside the fallopian tube, the egg begins its slow journey toward the uterus, propelled by a combination of ciliary motion and muscular contractions along the tube's walls. The timing of ovulation is crucial. The egg's lifespan after release is relatively short, about 12 to 24 hours. which means that sperm must be present in the reproductive tract during this period, or arrive soon after for fertilization to occur. This is why the fertile window of the menstrual cycle extends for several days before ovulation. Sperm introduced during this time can survive in the female reproductive tract for up to 5 days, awaiting the release of the egg. The overlap between sperm viability and egg availability is the narrow span in which conception is most likely. Ovulation also has effects beyond the ovary. The high estrogen levels leading up to ovulation influence the cervix to produce fertile cervical mucus, which is clear, stretchy, and alkaline, creating ideal conditions for sperm transport. The uterus, under the influence of estrogen, continues to thicken its lining in preparation for a possible pregnancy. Even the fallopian tubes respond to the hormonal environment, increasing their secretions to support and nourish both sperm and the egg during their meeting. After the egg is released, the ruptured follicle in the ovary transforms into the corpus luteum, a temporary glandular structure that begins producing progesterone. This hormone shifts the body into the luteal phase of the cycle, preparing the endometrium to support implantation should fertilization occur. If no fertilization takes place, the corpus luteum will eventually degenerate, leading to the drop in progesterone and estrogen that triggers menstruation. Ovulation is not always a perfectly symmetrical event between the two ovaries. In most cycles, only one ovary releases an egg, and the sides often alternate from month to month, though not with absolute predictability. In some cases, both ovaries release eggs in the same cycle, which can lead to fraternal twins if both eggs are fertilized. The process is also sensitive to external and internal factors. Stress, significant changes in weight, illness, or hormonal imbalances can delay or prevent ovulation, resulting in anovulatory cycles in which menstruation may still occur, but without the release of an egg. From an evolutionary perspective, ovulation represents a balance between energy, investment, and reproductive opportunity. Producing and releasing an egg requires significant biological resources, so the process is limited to a single occurrence per cycle in most cases. This measured approach ensures that the female reproductive system is not constantly expending energy on eggs that may not be fertilized, while still providing regular opportunities for conception. The precise timing also allows synchronization with environmental, social, and physical cues that may influence reproductive success. In human reproduction, ovulation is the moment when the pathways of the male and female systems converge. All the preparation that takes place in the male, the production, maturation, and delivery of sperm, aligns with the female system's single-day release of an egg. The success of fertilization depends on this coordination, a silent partnership between two complex systems that evolved separately, but now act in concert toward a shared biological purpose. In the next section, we will follow what happens immediately after ovulation, when fertilization occurs, exploring the moment when sperm and egg unite, the changes that take place within the oocyte, and the start of a new genetic combination that has the potential to grow into a living individual. This will connect the journey we have traced through both the male and female systems to the very beginning of embryonic development. 18. Fertilization, the union of sperm and egg. The union of sperm and egg. Fertilization is the pivotal point at which two separate lines of genetic material, one carried within the sperm, the other within the egg, merge into a single, unified blueprint for a new individual. It is the bridge between the independent journeys of the male and female reproductive systems, the instant when potential transforms into the first cell of a developing human. This process is both rapid and intricate, requiring precise timing, optimal conditions, and the completion of several critical steps that ensure the resulting zygote is viable. Although fertilization itself may occur in just a matter of minutes, the preparation leading up to it begins days, and even months, earlier. The event most often takes place in the ampulla, a widened section of the fallopian tube that lies between the tube's entrance near the ovary and its narrower midsection. This is a location where the environment is carefully balanced. The temperature, pH, and chemical composition are all suited to sustain both sperm and egg. The fallopian tube's lining produces gentle currents of fluid, carrying nutrients and signaling molecules that assist in the final maturation steps of sperm, known as capacitation. This process alters the sperm's outer membrane, making it more fluid and flexible, and increases the responsiveness of its tail to calcium, which enhances motility. Capacitation also primes the sperm for the acrosomal reaction, the release of enzymes from a specialized cap-like structure at the head of the sperm, without which penetration of the egg's protective barriers would be impossible. The egg awaiting fertilization is not a bare, unprotected cell. After its release during ovulation, it retains a coating of follicle cells called the corona radiata. These cells form a soft but substantial outer halo, still clinging to the egg from its time in the ovary. Beneath this lies the zona pellucida, a clear but firm glycoprotein layer that surrounds the egg's plasma membrane. The zona plays multiple roles. It protects the egg, facilitates species-specific sperm recognition, and later ensures that no more than one sperm can enter. When sperm reach the ampulla, they are met by this layered defense. Only those with optimal motility and membrane readiness from capacitation can pass through the corona radiata. They achieve this through vigorous tail movement and the gradual release of enzymes from the acrosome, which help to separate and digest the spaces between the follicle cells. The path to the zona pellucida is not direct. Sperm must navigate microscopic gaps, propelled forward in a steady but exhausting effort. Upon reaching the zona, the sperm encounter a finely tuned lock and key system. Specific receptors on the sperm surface recognize and bind to complementary structures on the zona. This binding is the trigger for the full acrosomal reaction, in which the acrosome releases a concentrated set of enzymes that bore a narrow tunnel through the zona's dense network. Multiple sperm may begin this process at the same time, but the journey through the zona is still a solitary one. Most sperm will fall short, losing energy or failing to penetrate deeply enough. When one sperm finally reaches the egg's plasma membrane, it binds to specialized microvilli on the egg's surface. The membranes of the sperm and egg fuse, allowing the sperm's nucleus and other essential structures to enter the egg's cytoplasm. This fusion instantly sets off the cortical reaction within the egg. Tiny vesicles just beneath the plasma membrane release their contents into the space between the membrane and the zona pellucida. These chemical changes cause the zona to harden and the sperm-binding sites to deactivate, creating an impermeable barrier to any additional sperm. This safeguard against polyspermy ensures that the developing zygote will have the correct number of chromosomes. Once the sperm's head is inside, its nuclear envelope dissolves, releasing its tightly packed DNA. The egg, which has been paused in the final stage of meiosis, now completes this division, discarding a small polar body and forming its own haploid nucleus. This female pronucleus moves toward the male pronucleus, drawn together by the egg's internal scaffolding of microtubules. As they approach, both pronuclei shed their membranes, allowing the chromosomes to intermingle. At this moment, the cell's genetic complement becomes a unified diploid set, half from the mother, half from the father, marking the true completion of fertilization. This newly formed zygote contains all the genetic instructions needed for building and guiding the development of a human being. The arrangement of genes determines inherited characteristics, from the broad strokes of height potential to the finer details of facial features. Alongside this genetic merging, a wave of metabolic activity sweeps through the egg's cytoplasm, activating the machinery needed for the first cell division. Within about 24 to 30 hours after fertilization, the zygote undergoes its first cleavage, splitting into two cells without increasing in overall size. This marks the beginning of embryonic development. The timing of fertilization is tightly constrained by the lifespan of the gametes. The egg remains viable for only about 12 to 24 hours after ovulation, while sperm can survive in the female reproductive tract for up to 5 days. This overlap creates a fertile window in which intercourse can lead to conception, even if it occurs several days before ovulation. The narrowness of this window underscores the precision required for successful reproduction. From a broader biological perspective, fertilization represents not just a union, but a culmination of selection. At every stage, barriers and checkpoints have filtered the sperm population, favoring those with optimal motility, structure, and timing. The cervical mucus, uterine contractions, immune responses, and the zona pellucida itself each act as stages in this selection process. By the time one sperm reaches the egg, it has already proven its ability to navigate these challenges. The egg, too, has undergone its own selective preparation, maturing over many days and being released only when hormonal conditions align with the readiness of the reproductive tract. After fertilization, the zygote does not remain in place. Gentle contractions of the fallopian tube's muscular walls, combined with the beating of cilia along its lining, guide the zygote toward the uterus. This journey will take several days, during which the zygote continues to divide from 2 cells to 4, then 8, and onward, becoming a compact cluster called a morula. By the time it reaches the uterus, it will have developed into a blastocyst, a hollow structure containing an inner cell mass that will form the embryo and an outer layer of cells that will contribute to the placenta. The successful union of sperm and egg is the foundation for all that follows. But it is not the end of the reproductive journey. The zygote must now find a secure place within the uterine lining, where it can implant and receive the nutrients and protection necessary for growth. This transition from a free-floating cell cluster to an anchored, developing embryo marks the start of pregnancy proper. In the next section, we will follow this critical journey into the uterus, exploring the stages of early embryonic development and the process of implantation. This will complete the link between the moment of fertilization and the establishment of a safe environment in which new life can unfold. 19. Early embryonic development and implantation. Once fertilization has taken place in the ampulla of the fallopian tube, the newly formed zygote begins a quiet but significant journey toward the uterus. This is a period of transformation and preparation, in which the single-celled zygote becomes a growing cluster of cells, steadily moving toward a destination that can offer nourishment, protection, and a stable environment for development. The journey lasts several days, and during this time, the embryo remains unattached, drawing on the resources stored within the egg while it undergoes its earliest stages of growth. The first stage following fertilization is cleavage, a process of repeated cell division without an increase in overall size. The zygote, still enclosed in the zona pellucida, divides into 2 cells, then 4, then 8, and so on. Each of these cells, known as blastomeres, becomes progressively smaller with each division, as the total volume of the embryo remains unchanged. This pattern of division allows the early embryo to produce many cells quickly, laying the groundwork for future specialization, while still maintaining the compact structure needed to navigate the fallopian tube. As the embryo moves along the tube, propelled by gentle muscular contractions and the coordinated beating of cilia lining the walls, it continues dividing. Around the 3rd day after fertilization, it reaches the morula stage, a solid ball of roughly 16 to 32 cells. The cells of the morula are tightly packed, connected by strong junctions that help maintain the structure's integrity. At this stage, the embryo still relies entirely on its own internal resources. There is no direct contact with the mother's bloodstream, and the uterine lining has not yet been engaged. By the 5th day, the morula transitions into the blastocyst stage. Fluid begins to accumulate inside the cluster of cells, creating a hollow cavity. This change marks the beginning of differentiation, the first step toward assigning specific roles to cells. The blastocyst consists of 2 distinct groups: the inner cell mass, which will develop into the embryo proper, and the outer layer of cells, called the trophoblast, which will form part of the placenta and other supporting structures. The zona pellucida, which has protected the embryo and prevented premature attachment during its journey, begins to thin at this stage, preparing for the moment when the blastocyst will hatch from it. The timing of the blastocyst's arrival in the uterus is crucial. The endometrium, the lining of the uterus, undergoes cyclical changes under the influence of estrogen and progesterone, becoming thick, glandular, and richly supplied with blood vessels during the luteal phase. This preparation creates a narrow window of receptivity, often called the implantation window, in which the uterine lining is most able to accept and support the developing embryo. If the blastocyst arrives too early, the lining may not be ready. If it arrives too late, the lining may already be beginning to break down in preparation for menstruation. When the blastocyst reaches the receptive uterus, it sheds the zona pellucida in a process known as hatching. This allows the outer trophoblast cells to make direct contact with the endometrium. Implantation begins with apposition, a loose alignment of the blastocyst against the uterine lining. The blastocyst typically settles in the upper part of the uterus, where blood supply is plentiful and conditions are favorable. Following apposition is adhesion, in which molecular interactions between the blastocyst's surface and the endometrial cells create a firm attachment. The trophoblast cells then begin to invade the endometrium in a stage called penetration. These cells secrete enzymes that gently dissolve small areas of the uterine lining, allowing the blastocyst to embed itself securely within the tissue. This process does not harm the uterus. Rather, it initiates the formation of a direct connection between the embryo and the maternal blood supply, which will be vital for continued development. As the trophoblast penetrates deeper, it differentiates into two layers: the cytotrophoblast, which remains as a cellular boundary, and the syncytiotrophoblast, a multinucleated layer that extends into the endometrium and begins forming the early placenta. The syncytiotrophoblast also produces human chorionic gonadotropin, HCG, the hormone that signals the corpus luteum in the ovary to continue producing progesterone. This maintenance of progesterone levels is essential, as it keeps the endometrium thick, stable, and rich in nutrients, preventing the onset of menstruation. Implantation is a gradual process, usually completed within 6 to 10 days after fertilization. Once securely anchored, the blastocyst is no longer a free-floating structure. It has established its first physical connection to the maternal environment. Nutrients and oxygen can now begin to diffuse from the mother's blood to the embryo, while waste products move in the opposite direction. This exchange will become increasingly sophisticated as the placenta develops, but even in these earliest stages, it marks the beginning of a partnership between mother and child that will continue throughout pregnancy. The success of implantation is a defining moment in reproduction. While fertilization creates the potential for new life, implantation allows that potential to be sustained. Without implantation, the embryo cannot receive the resources it needs, and development will halt. This is why the alignment of the embryo's readiness with the uterus's receptivity is so vital, and why the hormonal regulation of the menstrual cycle is so intricately tied to the timing of these events. From a biological standpoint, early embryonic development and implantation represent a balance between autonomy and dependence. The embryo begins as a self-contained system, relying on the stores within the egg and the processes initiated by fertilization. But within days, it must establish contact with the maternal system, integrating itself into a supportive environment while maintaining the genetic distinctness of its own cells. This integration is one of the most remarkable aspects of reproduction, as the mother's body accepts and nourishes a structure that is genetically half foreign. In the next section, we will explore the formation of the placenta and the development of the embryo in its earliest weeks. This will reveal how the initial connection made during implantation evolves into a complex support system capable of sustaining growth and development for the many months that follow. 20. The placenta and early pregnancy support. With implantation complete, the embryo transitions from relying on its own limited reserves to forming a partnership with the maternal body. This connection is made possible by the placenta, a remarkable organ that develops specifically for pregnancy and serves as the life support system for the growing embryo and later the fetus until birth. The placenta is not present at the moment of fertilization. Rather, it begins to form in the days following implantation, growing from the outer layer of cells of the blastocyst, known as the trophoblast. Over time, it becomes a complex interface that links two circulatory systems without ever allowing the direct mixing of blood. This separation is essential, as it allows the transfer of nutrients, oxygen, and waste products while preventing harmful immune reactions. The earliest stage of placental formation begins when the trophoblast differentiates into two layers: the cytotrophoblast, made of individual cells that maintain a structural boundary, and the syncytiotrophoblast, a multinucleated layer that aggressively, but precisely, invades the endometrial tissue. The syncytiotrophoblast expands into the uterine lining, breaking down small blood vessels and allowing maternal blood to flow into spaces known as lacunae. These pools of maternal blood create the first exchange areas, even before a fully developed placenta exists. By the end of the second week after fertilization, small projections called chorionic villi begin to extend from the cytotrophoblast into the maternal tissue. These villi greatly increase the surface area available for exchange between mother and embryo. Inside each villus, fetal blood vessels begin to form, connecting back to the embryo through the umbilical cord. As these vessels develop, they branch extensively, creating a dense network capable of absorbing nutrients and oxygen from the maternal blood in the lacunae, while releasing waste products such as carbon dioxide and urea. The placenta is more than just a transfer station. It is also a powerful endocrine organ. Very early in pregnancy, the syncytiotrophoblast produces human chorionic gonadotropin, HCG, the hormone that maintains the corpus luteum in the ovary. By ensuring that progesterone production continues, HCG keeps the endometrial lining thick and stable, preventing menstruation and supporting the developing embryo. As pregnancy progresses, the placenta itself takes over the production of progesterone and also produces estrogen. These hormones not only maintain the uterine lining but also help prepare the mother's body for the eventual demands of childbirth and lactation. The structure of the placenta is a testament to efficiency. On the maternal side, it connects deeply with the endometrium, anchoring itself securely while drawing from the rich blood supply of the uterine wall. On the fetal side, it connects directly to the embryo via the umbilical cord, which contains two arteries and one vein. The umbilical vein carries oxygenated, nutrient-rich blood from the placenta to the fetus, while the two arteries carry deoxygenated blood and waste products back to the placenta for disposal into the maternal circulation. This exchange happens continuously and quietly, without any conscious control, and is vital for every stage of growth. As the embryo grows, the placenta adapts. Its villi branch and multiply, increasing the area available for exchange. Its blood vessels expand to accommodate higher demands for oxygen and nutrients. The placenta also acts as a selective barrier, allowing many essential molecules to pass while blocking certain harmful substances. However, it is not impermeable. Some viruses, medications, and environmental toxins can cross it, which is why maternal health and environment remain important throughout pregnancy. The early weeks of placental support coincide with a period of rapid embryonic development. The inner cell mass of the blastocyst differentiates into the layers that will form all the tissues and organs of the body. During this time, the embryo is especially sensitive to its environment, as its organs and systems are in their earliest stages of formation. The placenta ensures that the embryo receives a constant, balanced supply of nutrients and oxygen to fuel these complex processes. Beyond its physical functions, the placenta plays a role in modulating the maternal immune system. Normally, the body's defenses would target and reject cells that are genetically different from its own. The placenta produces signals that help the mother's immune system tolerate the presence of the embryo, allowing pregnancy to continue without immune rejection. This delicate balance, protecting against harmful pathogens while accepting the genetically distinct embryo, is one of the placenta's most remarkable achievements. By the end of the first trimester, the placenta is fully formed and capable of sustaining the pregnancy on its own. The reliance on the corpus luteum fades, and hormonal production by the placenta maintains the stability of the uterine environment. The organ will continue to grow along with the fetus, eventually reaching a diameter of 15 to 20 centimeters and weighing around half a kilogram by the time of birth. Throughout pregnancy, the placenta remains the silent link between mother and child. It adjusts to the changing needs of the fetus, increases its efficiency as demands grow, and serves as a record of the pregnancy's history. When the time for birth arrives, the placenta's role is complete. After the baby is delivered, the placenta detaches from the uterine wall and is expelled in a process known as the afterbirth. While its physical presence is temporary, its influence lasts a lifetime, as the nutrients and oxygen it provided have shaped every stage of fetal growth. From the moment of implantation to the final moments of pregnancy, the placenta is a central player in human reproduction. It is both a physical and biochemical bridge, a protector and provider, and a mediator between two genetically distinct organisms. Without it, the intricate dance of fertilization, development, and birth could not proceed. It stands as the final piece of the reproductive story. We have traced the culmination of the male and female system's efforts and the secure beginning of a new life.